Hello everyone, welcome to my channel. Today we will discuss about the new concept that is spermatogenesis. Okay, we will start here with contents. What we'll discuss here, like some points, I will cover definition of spermatogenesis, stages of spermatogenesis process, and three phases in formation of spermatid. These three concepts I will cover. Okay, we will see one by one. Before going to definition, we will learn about introduction. Okay, spermatogenesis introduction. Spermatogenesis is the process by which the seminiferous tubules of the testis produce haploid sperm. Here, it is the process of formation of sperms. Sperms are in haploid number. Okay, we seen in seminiferous tubules. Okay, next, diploid cells like nothing but 46 chromosomes. Okay, 46 chromosomes go through meiosis to produce 4 haploid sperm cells. Here 46 will be divided to form 23 chromosomes that we call as the haploid cells. That cells we call as the haploid cells. Each sperm cell is unique and different from the parent cell and the other sperm cell. Here, each sperm cell is very unique and different from the parent cell to another sperm cell. Okay, next. Definition. See your definition. The formation of spermatozoa or sperms in the testis is called spermatogenesis. Here, spermatozoa or sperm formation we see in the testis. This is testis. Okay. This process we call as the spermatogenesis. Okay. A pair of testes are found in males. It is found in males. Okay. It is attached to the connective tissue. Dorsal body wall by the connective tissue membrane is called mesorchium. Mesorchium. Okay. The testis is formed of thousands of seminiferous tubules here. Seminiferous tubules. These are very minute tubules. Okay. And they lead into vasa defensia. They leads into vasa defensia. Seminiferous tubules are separated by the interstitial cells. These are separated by the interstitial cells. See in this picture. Like transverse section of seminiferous tubules. The interstitial cells are separated by the seminiferous tubules. Okay, and each seminiferous tubules is covered by the basement membrane. This basement membrane and lined with germinal epithelium, and the germinal epithelium cells are separated by the gained cells. Gained cells. That gained cells we call as the Sertoli cells. These are gained cells, this and this. Okay, these long tube-like structures we call as the gained cell. That cell we call as Sertoli cells. Okay, the germinal epithelial cells develop into the spermatozoa, and Sertoli cells we seen in Sertoli cells gives to nourishment for the development of the spermatozoa or spermatids. Okay, next. The process of spermatogenesis has two stages. Here, two stages we will discuss. The formation of spermatid and spermiogenesis. Okay, next. First one, formation of spermatid. See here, spermatid is formed from the epithelial cells of seminiferous tubules. We already know about that. epithelial cells we seen in testes. Okay, in there spermatid cells will be formed. Next, the germinal cells which develop into spermatids are called primordial germ cells. Here, see in this picture, primordial germ cells. Germinal cells which develop into the primordial germ cell. Okay, next. There are three phases in the conversion of primordial germ cells into spermatids, like multiplication phase, growth phase and maturation phase. These three are very important in this concept. Now we see one by one. Multiplication phase. Here, primordial cells are large in size and their nuclei are distinct. See in this picture, primordial cells are large in size. Okay, and nucleus will be distinct. It will be small. And meiotic division takes place here. In this, we see meiotic cell division. Okay, the resulting cells are called spermatogonia. Spermatogonia or sperm mother cell we call as the sperm mother cell. Okay, each spermatogonium has a diploid number of chromosomes, 2n number, like 46 chromosomes. Next, growth phase. Here during this phase has a diploid number chromosomes increases or will be increases compared to this and this. See, it will be increases. Primary spermatocytes are we seen. Is it? Yes. These have diploid numbers also. Okay. And diploid cells 2n in number. Next. Resulting cells spermatogonia or sperm mother cells. Here also we call as the sperm mother cell or spermatogonia. And diploid number of chromosomes we seen here. Diploid numbers 2n or 46 chromosomes. Next, maturation phase. Each cell divides by meiosis. Here we seen meiosis division. In previous two stages we seen mitotic division. Okay. Meiosis consists of two divisions, like first meiotic division. Here we seen haploid numbers of the cells like 23 chromosomes. Okay. And second meiotic cell division, here it will be undergoes spermatocyte. It will be divided into two cells. That cells we call as the spermatids. Okay. See in this picture, two cells will be formed. Again it will be divided in undergoes or according to meiosis style division, 4 will be formed. These are haploid in number or haploid in number. Next, second one is Spermiogenesis. Okay. See, the transformation of the spermatid into spermatozoon or spermatozoa is called as the Spermiogenesis. Here, formation of the spermatid into spermatozoa or sperms is called as the Spermiogenesis. Next, in this process we seen some differentiations, some changes occurs in some organelles. Like organelles nothing but in nucleus, mitochondria, Golgi complex, vacuoles, etc. Okay, we seen some changes. Like first one, nucleus gradually diminishes in its size and losing water. Here nucleus will lose water and its size will be changes. Okay. Next, chromosomes become concentrated and are closely packed. Here chromosomes will be concentrated and it will become closely packed like structure, and all the achromatic substances, nucleus and RNA disappear. Here we can see like nucleus and some substances and RNA. Next, the nucleus becomes elongate. Here nucleus become elongate or cap-like structure. The Golgi complex develop into the acrosome. Here Golgi complex will be convert or develop into the acrosome. See in this picture, acrosome. The small vacuoles gradually fuse together to form large vacuoles. Small vacuoles or granules to combine or fuse together to form large vacuole. This process we call as the acroblast. This process we call as the acroblast. Next, the acrosome and the nucleus together constitute the head. See here, the acrosome and nucleus. The acrosome and nucleus to form head portion of the sperm. Next, the distal centriole develops a filament-like structure called axial filament. Here, distal centriole. See in this point, 3 points are there in the last point, that we call as the distal centriole. Develops thread-like structure or filament, we call as the filament-like structure, that we call as the axial filament. Okay. Next, mitochondria of the spermatid are aggregated together to form a large mass in the region of the centrioles. This process we call as the mitochondrial cloud. These are mitochondrial cloud. Okay, here spermatids are aggregate together to form a mass of the centriole region. That region we call as the mitochondrial cloud. Okay. Next, the cytoplasm flows backwards and forms a thin layer around the nucleus, middle piece and tail. Here we see the cytoplasm will be move or flow backward direction or backwards, and it forms thin layer around the nucleus. That seen in their tail like structure and middle piece. Okay, middle piece. Okay, this is about spermatogenesis. Okay, overview we seen here. There are main 3 phases are there, like multiplication phase, growth phase, maturation phase. In multiplication phase, we seen meiotic cell division, and growth phase, meiosis cell division. Here, first and second meiosis we see, and maturation phase, haploid number of the spermatids are present. Okay. I hope you understand this video. Thank you. Please subscribe my channel, like it and share it.